Hello everyone. In this class, we'll try to learn about some of the properties and classification of the nerve fibers. Some of the properties of nerve fibers are excitability, conductivity, unfatigability, refractory period, all or none response, summation, and accommodation. So some of the properties of nerve fibers we have discussed in the previous class in neuron, such as refractory period, all or none phenomena. So in this class, we are going to uh, study the other properties of nerve fibers. Excitability. Excitability is the property by virtue of which cells or tissues respond to changes in the external or internal environments. The nerve fibers are highly excitable tissues. They respond to mechanical, thermal, chemical, or electrical stimuli. In an experiment setup, electrical stimulus is usually employed because of its strength and frequency can be accurately controlled. The factors that affecting excitability are strength and duration of the stimulus that we have learned in strength duration curve in the previous class. Then it also depends on the extracellular calcium. Decrease in the EC of calcium increase the excitability of neuron by decreasing the threshold potential, and increase in the EC of calcium stabilizes the membrane by decreasing excitability. Conductivity. On stimulation, action potential is generated in the nerve fiber, which is propagated along its entire length to the axon terminal. What is orthodromic and antidromic conduction? Impulses normally pass from synaptic junction to the axon terminal, which is called as orthodromic conduction. Conduction in the opposite direction is called as antidromic conduction. Axon reflects an example for antidromic conduction. If action potential is initiated in the middle part of the axon, it can travel in both the directions. Thus, axon can conduct in either direction. Summation. Application of subthreshold stimulus does not evoke an action potential. However, if subthreshold stimuli are applied in a rapid succession, they are summated and they produce an action potential. So this property is called as summation. Accommodation. Application of continuous stimuli may decrease the excitability of the nerve fiber. This phenomenon is called as accommodation. If a nerve is submitted to the passage of constant strength of current, the site of stimulation shows decreased excitability, decrease in the excitability. The accommodation consists of a rise in threshold of the tissue during stimulation. A similar feature observed in the nerve endings is called as adaptation. Thus, nerve fiber accommodates while the nerve endings adapt. Unfatigability. Nerve fiber cannot be fatigued even when they are stimulated simultaneously. So this is because the nerve fibers primarily conduct impulses, that is propagation of the action potential, that do not involve expenditure of energy. Coming to classification of nerve fibers can be classified based on their function as a motor, sensory, or secretomotor. Based on the myelination, they can be myelinated or unmyelinated, and based on the diameter and conduction velocity, the nerve fibers have been classified according to Erlanger and Gasser classification. So this classification is divided into divides the nerve fibers into A, B, and C type of nerve fibers, where A further divided into A alpha, A beta, A gamma, and A delta. Depending on the fiber diameter and velocity of conduction, they have been classified accordingly. So type alpha fibers are seen in a somatomotor and proprioceptive fibers, whereas A beta are touch and pressure fibers, whereas A gamma they are motor to the spindle, A delta they are pain and cold and touch fibers, whereas B forms the autonomic preganglionic fibers, and C fibers therefore somatic sensation, and C sympathetic fibers they form 4 postganglionic sympathetic fibers. They've been classified based on the numerical classification. So this is a sensory classification. The types are 1A, 1B, type 2 fibers, type 3 fibers, and type 4 fibers, and they are synonymous with A alpha, A beta, A gamma, A delta, and C fibers of the Erlanger-Grasser classification. Okay. So these are purely sensory. 1A type of fibers, they are primary spindle afferent from the annulospirings, and IB fibers are from the Golgi tendon organ, and type 2 fibers are secondary spindle afferent from flowers spray ending and also from the touch and pressure receptors. Type 3 are pain and cold receptors, and type 4 for pain and temperature. Some of the demyelinating diseases, as we see in multiple sclerosis, where you can see the patchy loss of axonal myelin, which is going to occur at the several areas of the nervous system, and which leads to decreased conduction velocity of the nerve fibers. Some of the pathways which commonly affected are pyramidal tracts, cerebellar tracts, medial longitudinal fasciculus, optic nerve, and posterior columns. In multiple sclerosis, usually symptoms are there will be sensory abnormalities, decrease or abnormal sensation or pain. There can be muscle weakness, fatigue, spasticity, optic neuritis, diplopia, or ataxia. In demyelinating form of Guillain-Barré syndrome, where there will be loss of myelin causes the abnormal conductions. Coming to nerve injury, when nerve is sectioned, the distal part of the nerve separated from the cell body, which is going to degenerate, and this is called as Wallerian degeneration. The proximal portion of the axon still attached to the cell body then develops a growth cone and grows distally toward the distal portion, and the changes associated are known as regenerative changes. The soma gets informed about the injury by absence of substances transported retrogradely from the axon terminals. There can be different gradings based on the severity. Nerve injury is typed into 5 degrees: first degree, second degree, third degree, fourth degree, and fifth degree. The first degree is transient loss of function that occurs due to the mild pressure on the nerve, which is caused by local ischemia following obstruction of the blood flow. The nerve fibers completely recover within few hours to few weeks. Then second degree injury, severe nerve damage with intact endoneural tube occurs. So these type of injuries occurs due to the severe and prolonged pressure on the nerve, and regeneration is delayed but complete as endoneural tube is intact. The third degree injury occurs because of the severe damage to the nerve fiber that interrupts the endoneural tube, where the fourth degree injury occurs because of the severe damage to the nerve, and there will be disorganization of the nerve fasciculi. And fifth degree injury is a complete transaction of the nerve, and you can see degenerative changes that occurs quite early. Following injury, so these are some of the changes that you can see in the nerve fibers. There can be degenerative changes and regenerative changes. Regenerative changes, they're going to affect the transmission of the impulse. There can be axonal changes. Then the effect of stimulation is altered, and there can be changes in the soma and the stum. Okay. Whereas regenerative changes can be studied as axonal changes, somatic changes, and changes that occurs in the target structure. The degenerative changes, Wallerian degeneration. August Waller discovered the degenerative changes occurring in the distal segment of the axon. So here, shortly after the injury, synaptic transmission occurring at the axon terminal stops within hours. Then the axonal changes, where axon swells and the terminals retract from the postsynaptic target. Gradually, over a period of several weeks, the axon and its terminals degenerate. If the axon is myelinated, round fatty enlargements form all over the myelin sheath that looks like series of beads. The myelin sheath breaks down, but the myelinating cells remain viable. The debris created by the disintegration of the axon and its myelin is devoured by the macrophages and Schwann cells in the peripheral nervous system and microglia in the central nervous system. The phagocytic process is rapid in the peripheral nervous system than in the central nervous system. And effect of stimulation. On stimulation, distal axon can conduct an action potential up to 3 days, but this ability is impaired from 3rd to 5th day. The action potential cannot be initiated after 5th day. Changes in the soma and stump are they are known as retrograde degeneration. Okay. So in retrograde degeneration, cell body swells and become rounded. The nucleus also swells and moves to an eccentric position. Endoplasmic reticulum, instead of being closer to the nucleus, reassembles around the periphery of the cell body. The ribosomes appear disorganized and Nissel's granules gradually disintegrate and they are stained weakly with the basic dyes. Okay, so this is known as chromatolysis. There can also be regenerative changes. The soma tries to repair the axon by synthesizing new structural proteins that fills up and distance the cisterns of the rough endoplasmic reticulum, and this is known as axonal reaction. Chromatolysis is reversible if the neuron survives and reestablishes its contact with the appropriate target. During regeneration, so these are the changes that you can see in the axon, soma, and in the target structure. The axonal changes are the proximal end of the axon develops mini sprouts. The sprouts elongate by formation of the growth cone at their growing end. The Schwann cells that had survived the degeneration multiply and forms rows along with the pathway of previously taken by the disintegrated distal axon. Out of the many sprouting branches, one branch finds the way through the Schwann cells and they finally re-innervates the original target structure. Okay. So this is the site at which there is injury and the distal part gradually is going to degenerate. Okay. And the proximal part, they are going, because they are in touch with the soma, so they survive. Okay. And with the Valerian degeneration, many structural changes that you can see. Okay. But the Schwann cells, they are going to remain, and then the regeneration is going to occur if the neural lemma is going to remain contact after the proximal stump develops mini sprouts, and one of the sprout is going to get a direction into the Schwann cells, and it is going to cause the regeneration of the disrupted neuron. The regeneration rate of the growing axon is about 1 to 4 millimeters per day. The Schwann cells then lay down their bilayered membrane to form myelin sheath around the newly formed axon. Somatic changes, the soma regains its size, the number of nasal granules slowly reappears, the other organelles also appear, the cell loses excess fluid and regains its normal size, the nucleus occupies the central position. The changes in the target structure are neurotransmitter release at the axonal terminal decreases. The decreased neurochemical at the synaptic cleft leads up to upregulation of the receptors in the target structure. Therefore, when neurotransmitter is released following regeneration, the response of the target tissue to the neurotransmitter is increased, and this is called as denervation hypersensitivity. So this is another diagram showing the injury, the Valerian degeneration distal to the cut, and then the regeneration process where they are going to form different sprout, and one of the sprout is going to elongate into the distal regenerated tube, and it helps in the regeneration of the distal part of the cut. Okay. And then the synaptic connectivity is re-established. The factors which are influencing regeneration. The successful axonal and somatic regeneration depends mainly on 4 factors, that is severity of the injury, condition of the soma, location of the injury, and neurotrophins. If the gap between the proximal and distal stump is more than 3 millimeters, the multiple outgrowths intermesh and form a tumor-like swelling which is called as neuroma. If neuroma is formed, successful regeneration can never occur. If the neuroma involves the sensory fibers, the pain is felt at the site when touched. Then the condition of the soma. If the axonal damage is close to the cell body, a larger part of the neuronal membrane and cytoplasm is lost. The neuron often dies instead of regenerating. Then the location of the injury. In the CNS, single oligodendria sends out mini process to myelinate around 15 axons. Therefore, even though the axonal sprouting occurs, the oligodendroglia cannot form path along which sprouts can grow. Besides, glial scars formed by the astrocytes pose obstruction in the pathway of the growth cone. Hence, axons in the CNS regenerate less successfully than axons in the peripheral nervous system. Then it all depends on neurotrophins. When specific neurotrophins are administered to the site of injury, the growth of regenerating sprout is enhanced. And enmeshing of the branches does not occur because of the neurotrophins. Some of the factors which promotes the neuronal growth are neurotrophins and other growth factors like neurotrophins are nerve growth factors, brain-derived neurotrophic factors, neurotrophin 3, 4, and 5, whereas there can be ciliary NTF, glial cell derived neurotrophic factor, leukemia inhibitor factor, and factors like FGP, platelet derived growth factor, tissue growth factor, and insulin like growth factor 1. They play important role in promoting neuronal growth. So this is in brief about some of the properties of neurons, regeneration and degeneration of the neuron. Thank you.